Major support for Do the Math has been provided by Chevron, the Kern County Superintendent of Schools, Edison International, Valley Strong Credit Union, California Resources Corporation, Bakersfield West Rotary Stroop Family Foundation, Panama Buena Vista Union School District, Bakersfield City School District, Kern High School District, and AC Electric Company, with additional production assistance provided by these supporters of education in Kern County and throughout the state of California.
We're here at Taft College today. We're in the lab. We're on campus. We've got an amazing crew of students helping us out today. And today we're going to talk with Professor of Life Science here, Greg Galling. Greg, thank you very much for having us in your lab today. We have a really exciting experiment that may share some relevance as far as the way that our viewers may interact with the world today.
Yeah. Basically what we're going to do, we do sort of a hand washing experiment, but we're going to look at the bacteria that are kind of currently on your fingers, things you've picked up through the day, go through a pretty rigorous hand wash, and then it's sort of looking at how well did we clean our hands off, but there's also some other lessons we'll learn from this, the kind of interesting results we get in the end.
Now, a lot of kids are going to look and they're going to brush off this idea, I don't got to wash my hands, I don't see anything on there, right? In full transparency, about 45 minutes ago when I left my house to come here today, I put some hand sanitizer on. So the only things that I've been able to interact with my hands have been the steering wheel inside my car, maybe opening up some handles. But the idea is just because you see it doesn't necessarily mean it's there. And what this does is bring that into vision.
Yeah, bacteria, you can't really see them with the naked eye, but they're all over us. In the average person, depending on their size, you're looking at maybe 10 trillion, 100 trillion bacterial cells in and on us. So it's just, it's a great mystery. Just because you can't see it doesn't mean it isn't there.
So we've got some dishes over here that we're going to utilize, and kind of walk us through this experiment and what we're going to do.
So what we have, it's just it's a petri dish and it's got a nutrient auger. So there's lots of things that bacteria like to eat, and they'll grow on top of the surface. Kind of like if you've had you leave bread out or fruit out for a couple of weeks and you start to see the little, green mossy stuff growing on it, except we're looking at bacteria instead of fungi. So what we'll do is we're going to take a look at what's on your hands now, sort of a pre-washing. We'll wash our hands and then we'll take a look with 3 fingers and see what's on after we wash our hands.
So this is going to be kind of a before and after of filth on my fingertips.
Pretty much.
Okay.
Yeah.
Well, let's go ahead and I believe our producer Aaron's going to be joining us on this experiment as well.
Yeah, so if you want to go ahead and set up your plates, just kind of draw a line. I've got a Sharpie, I can get another one, and just draw a line down, one side write pre, the other side post, and maybe your initials. And then just with your 3 fingers on your right hand, just kind of gently touch them to the petri dish. And it feels just like Jell-O, okay? And then we'll put the lid back on, wash our hands, and once they're dry, then we'll go ahead and touch the post side.
Okay. So Aaron, go ahead and take care of yours. I'll go ahead and start the press here. All right. You can't see the lines through there. This was the pre-side, so I'll go ahead. Does it have to be a certain 3 fingers? Can it be pinky to middle? Can it be pointer to ring?
I'd get the index in there because that's one you tend to use a lot.
Sure is. All right. 3, 2, 1. Just a little press. Didn't have to be too hard there. Enough to see the fingerprints. Okay, so we've got the 3 fingers in there, and now the next part of this is 2 minutes of hand washing, plus about 45 seconds of drip.
Yeah, just let it air dry. If we use a paper towels aren't sterile, so you could be spreading something else on it. And I'm just going to cover these up. Again, the whole unseen world out there. Right now, there are bacteria falling. We don't know it, and some could have landed on here. But we'll know in the end if any did or not. But so we've got a couple of sinks here, some soap dispensers. I have them get kind of fill up about 4 or 5 pumps of the soap, and I'll turn the water on for you. better if it's a little bit warm, but go ahead and just really rub the heck out of those. Try to get any traces of fingerprints off. I don't know if you want to use that sink or...
Well, somebody's counting to 120 here, so I know when to start.
Yeah. All right, so we'll say that's been about 15 seconds.
As enthralling television as it is watching me wash my hands, we're going to go ahead and come back, and for our next break, we're going to go ahead and throw these into the incubator. We're going to talk a little bit about what that does, and then we're going to get a look and see exactly down the line what this looks like as we skip forward through time. Back here at Taft College, if you missed our first segment, we have some explaining to do. We washed our hands. We're here at Taft College with Professor of Life Sciences, Greg Golig. And we did a Petri dish experiment. We're exploring bacteria, the unseen invader. We did a pre-gel where we took our fingertips, pushed them in to see what would be there, and then we washed our hands to see what's left afterwards to kind of illustrate the importance of hand washing. So what we've been doing is we have been letting our hands kind of dry out. Aaron as well is joining us on this experiment. We're just about there where we're no longer dripping, and Greg, we're ready for the next step.
Okay, so what we'll do, you've got your Petri dishes. You've already taken your 3 fingers and just touched them to the pre-side. Now you're just going to repeat with the same 3 fingers on the post side. Okay.
All right, so, and as soon as we do that, as we learned from our last segment, we want to put that cap back on.
Yep.
So we don't let any unseen bacteria from the air around us come back through there. Okay.
Yep. All right. And then what we would do now, so the plates are done, you would have deposited single cells of bacteria on the plate, and depending on the type of bacteria, they take about 20 minutes to divide. So every 20 minutes they go to 2, then to 4, and after about 18 to 24 hours, you could see this little mound of cells on the plate. And I have some of those if you want to take a look at different types of bacteria. So we'll put these in an incubator.
So tell us a little bit about the incubator, the conditions inside, and how that facilitates the growth of the bacteria inside the dishes.
Right, it's an excellent question. So the bacteria that hang around our bodies that cause disease, there's obviously things about us that they like, the food, the nutrients, and one other thing they like is the temperature. So, like, just like with all of us, there are certain temperatures we kind of prefer to be at, and same thing with bacteria. So this is set basically to body temperature, around 36 degrees Celsius. And so we'll just put them in here, and these are some examples. Pull out this one. I've moved some of the bacteria around, but you can see this is about a day old. And this bacteria is called Bacillus subtilis, and it's something that you can find actually. I don't know if you want to smell it, maybe you don't.
I'm good.
Yeah, if you did, it's going to smell kind of like rotten feet, and this is part of the reason why
I already got those.
you get foot odor. So anyway, that's what you'd look at after about 24 hours of growth. You put your stuff on, you don't see anything. 24 hours later, boom, there's these huge colonies.
So if we could estimate how many, we could use the amount that we see there to kind of estimate how many were there to begin with, kind of working backwards.
Well, we know each one of those little colonies, it starts off as exactly one cell. And as it divides, it's sort of like if you had a bucket of sand and you started pouring it out, and it starts to build up and spread out and build up and spread out. And what's kind of neat is each species of bacteria, they've got a particular way, like the little pigs, a way that they build their house. Some like to build kind of mounds, others are kind of flat. The colors are different between them. Some are really tiny colonies, others are kind of large and taking over the whole plate. And it's interesting because, again, each species has a particular way they all grow, they make their colony, they make their house in that same manner.
So it's not just about and it's not just about the fact that it's doubling, but how rapidly it's doubling that is going to let us know how aggressive some of these bacteria are in certain conditions.
Right. And those colonies that you looked at, you've got somewhere on the order of hundreds of millions to a billion cells in that colony. They're that small.
So I understand that our students here have taken part of this activity yesterday.
Yes. Yeah. So you guys ready to see your results? You excited or nervous? I'm not really sure.
Hey, quick survey. Who's going to have the winner for the most full bacteria dish here? You don't have to point fingers.
Yeah,
We're
don't
rolling
point
here.
at me.
You know the answer.
All right. So I'm going to take those out.
So when we come back, we're going to reveal the winner of our events from our tests from yesterday to see how rapidly just a small sampling of these just in one day can have as far as an impact on the growth of these bacteria. Back here at Taft College at the biology lab here with Greg Galling, he's a professor of life sciences. We've been doing a bacteria experiment, pressing fingers on petri dishes, letting them incubate for a day, and seeing what comes up before and after we wash our hands. The students here in this class took part in this yesterday. They're collecting their dishes from a day in the past. Let's see how this turned out for everybody.
All right.
Let's take a look here, starting off. Sir, your name, please?
Ahmed.
Ahmed. Thanks for joining us, Ahmed, and thank you for sharing your bacteria with us. So, let's take a look. Tell us some of your observations here.
So this side has a lot more bacteria than this side. I'm guessing that this side was the pre-side and this is the post-side because I learned earlier that actually when you scrub your hands a lot, it reveals some of the natural bacteria that we have on them. It's called normal flora. So when you scrub it a lot, then a lot of that bacteria will show. And not all bacteria is bad. So that bacteria is actually good for us. So we see here on the post side, after scrubbing, it exposed it and it let it grow a lot. So I'm guessing that this side here was pre and that's post.
That's a lot of confidence to say, hey, my hands are cleaner before I wash my hands.
Hopefully.
To be able to say that. That's fascinating. Is that what you thought was going to happen?
I think so. I've done this lab in a different class and I got sort of similar results. I'm hoping that this is the post side, but if it's the pre side, that's probably bad news.
That's right. Well, let's see if anybody's got some bad news here.
Well, I can't say too much about mine. I mean, this is my pre-side, and they look about the same,
Well,
so
that's interesting.
I guess...
One thing when you're looking at them, again, each bacterial species has a different way that it grows, different colors and sizes. Look and see on your post, do those look like a lot of different types of colonies? Do you have more different colonies on the pre?
Well, seems like there could be a different colony that did come out after hand washing
And you can
compared.
observe that just by looking at the size of the colonies, the colors, the shapes.
So it seems like they have about the same mound, like Professor Goling was saying. They can spread out, they can grow in heights or in masses. It seems like there's just a few of the same sizes and they all have the same color, it appears.
And what's interesting is you use the same three fingers, but on the pre, different mounds are growing larger than on, let's say, the post. So like we could see different clusters on different fingers, and you use the same three fingers when you press down. So that's really interesting. Who else can we share with?
I saw some good ones earlier that went from hardly anything on the pre to quite dirty on the post. Anybody willing to share? We could black out faces, I guess. It's okay.
Okay.
So let's take a look.
Oh, that's a good one.
Oh, wow.
That's awesome. Yeah, I wouldn't want to talk either. So what really stands out when we look at this?
Well, you know, when we do this with students, if they haven't done it before, they're always amazed that, and they think they messed up. They think, oh, I must have put my post hand on the pre side, you know, switched them around. And they just can't believe that their hands would have gotten dirtier from washing them, because that's what we're always taught. Hey, well, clean your hands, wash your hands. And, you know, the first thing they think, oh, it must be the water at Taft College, that's it. You know, this is the water, this is good water. This, you know, it would be criminal if we had that much bacteria in the water here, and we don't. It's good. And what a lot of students and people in general don't realize, what's happening is, on the pre-side, that's everything that you've touched during your day, your steering wheel, for instance, and you're just transferring those bacterial cells onto the plate. When you wash your hands, you're rubbing off some of the skin cells, and those are getting rinsed down the drain, and there's just this jungle of life underneath those skin cells. Again, we've got, you know, the average human body maybe has 1 trillion human cells, 1 to 10 trillion, and we've got about 10 times that amount of bacterial cells in us, on us. So what you see in the post, this is what we'd call normal flora. This is stuff that we all have. It's generally speaking not pathogenic, and in many cases it's beneficial to us. It can prevent other organisms, certain pathogens, from actually growing. They're all trying to compete against each other for space, for nutrients, and if they've adapted to live on our fingers, that gives them, you know, sort of a leg up, so to speak, with that.
The message isn't necessarily don't wash your hands.
Right.
The message is when you do, some of the healthier, more natural bacteria that's already in you is exposed, and you can kind of see what's there.
Yeah, and if that gets on you or somebody else or your sandwich that you're eating, it's it's already part of you, so it's not necessarily a bad thing. Yeah.
Well, Greg, thank you so much for having us here to share our appreciation for allowing us in here at Taft College. We wanted to grant you some of our traditional do the math, congratulatory tile. Post that, place that wherever you'd like. Put it on display where you can.
Thank you very much.
Hey, thank you very much, everyone. That'll do it here from Taft College. Until next time.